Thank you, Dr. Park, for that kind introduction and the invitation to speak. A few disclaimers up here. This is where I work at the University of Chicago. It's indeed a great honor to be here in this beautiful city, in this beautiful venue, amongst so many friends that I've had over the year through the Society of Critical Care Medicine. These are the people that did some of the work, maybe all of the work, from my laboratory and some of the funding that we've received. A particular shout out to Dr. Olga Zabarina, who is the microbiologist that runs the lab and is sort of the lifeblood of this laboratory. It's again a great honor to be the Max Harry WEIL professor. I read a lot about Dr. WEIL when I was a resident and a critical care fellow, and he is certainly a pioneer in the field. It's an honor to have my name associated with him in this venue. I also wanted to point out my homies at the Surgical Infection Society. This is the group of people, many of whom you may know because they've been past president of this society: Pam Lipsett, Phil Berry, John Marshall, who were presidents of the Surgical Infection Society and president of SCCM. The constant encouragement by this group of people, including Lynn Heido, who's in the audience also, has been really something that has made me pursue basic science to study infection and surgery all these years. Now I'm going to take you down a path of thinking that might anger a few of you in the audience. I want to especially thank Timothy Buckman, who encouraged me to write all this up in a manuscript and submit it to Critical Care Medicine, and it's actually in print. Just a few days ago, I saw it pop up on PubMed, and thank you again for that opportunity. And of course, Craig Coopersmith, your past president last year, who has been a constant inspiration to me to move this field forward. But when we think about critical illness and we think about the pathophysiology of critical illness, it's highly skewed toward immunology. And even in the new definition of sepsis, where we say microbes initiate this disorder, but we really believe they become irrelevant once antibiotics are initiated at the right time and the right antibiotics, and source control is achieved. And so if you read anything about sort of the immunopathology of sepsis or the pathobiology of sepsis or the pathogenesis of sepsis, it really does focus on the immune system. And many papers will state right up front that it's an inflammatory and immune disorder primarily, and that's what drives the organ failure and death, and there's no question that the immune system is critical. But I think we've forgotten about the microbes that remain hidden in microscopic habitats within the body that still play a role in driving the disorder, and that's what I want to talk to you about. Now, the promissory note of all of the immunologic work that's been done in the field of critical illness, the promissory note has been we can achieve immune control at each time point across the entire course of critical illness if we just understand what the immune system is doing at each time point. If we just drill down and get a granular understanding, we can create a motherboard in which we can turn cytokines up and down, and I've saved a little spot here for the next cytokine that's going to show up, and we can instantiate that knowledge and antibodies or upregulation and downregulation into the patient, and it's going to be huge, and we can do it. And much of the reason that that has occurred is because this sketch, which has appeared in many articles, talks about how initially patients are injured or infected and there's immune activation, and of course we get SIRS, and then that leads to organ failure. And now we've done an excellent job due to the sepsis campaigns from this organization and others and early resuscitation that we're now seeing this problem of late onset sepsis. So first, it was thought we have too much inflammation. Now the term immunoparalysis has come up. Myeloid suppressor cells are important, and that's causing late onset sepsis, which is now the most common cause of sepsis and death from sepsis in intensive care units in developed countries. And what we've been somewhat dismissive of is the stochasticity, the sort of perturbances, as we heard this morning, that can occur. These multiple takebacks to the operating room, prolonged exposure to antibiotics, occult hits, the kind of nutrients that we use, are they adequate when they go in this bioreactor now of the microbiome? How are they being processed? Where do they end up? Are we giving the right things? It's not just about nitrogen balance, it's about good quality food that feeds the entire superorganism made up of bacteria and host cells. And so this problem has cropped up where a common cause of death now in an intensive care unit is withdrawal of care. Now, I want to propose a more balanced view of this in the context of the microbiome. And I want you to understand that the presence of microbiota on your epithelial surfaces, the most dense of which is in the gut, plays a critical role in the tonic stimulation of the immune system. And when that microbiome shifts to a pathobiome, where you've eliminated the normal health-promoting microbiota and you now have a preponderance of pathogens colonizing those surfaces, you can get a pathoadaptive immune response. And the extent to which loss of the microbiome and the presence of the pathobiome drives this response is not fully characterized yet, but is coming to light. And a general hypothesis is that if you maintain a healthy microbiome, you can drive what I call a recovery-directed immune response. But what is a healthy microbiome? What do you exactly mean by a recovery-directed immune response? Is that Th1 cells, Th2, Th17, IL-22? What is it? Well, it's a lot of things, and it can't just be identified as a single cytokine, because we've been down that road the past 20 years. And of course, the problem is, over the course of critical illness, we do a really good job of getting rid of the microbiota and disturbing them with what I call unavoidable exposures in the ICU. Nobody's interested in not giving PPIs or taking a patient with severe acute pancreatitis who looks really sick and saying, let's not give antibiotics, et cetera, et cetera. But what happens is that competitive exclusion of pathogens becomes disabled by these processes. And it's not just a large biomass of normal bacteria that needs to be just placed in there. That biomass of normal microbiota are doing far many more things than we actually understand at this point. The other problem is much of what we know about the immunopathology of sepsis, we know from this model. In fact, I erased the authors of this to protect the innocent. Many of them are outstanding immunologists who have advanced our understanding of the immunopathology of sepsis, and they should be commended for that. But this is a model in which you ligate the cecum, puncture it, cause dead gut to be left in the abdomen, and you don't give antibiotics, and you don't do any of the other things there. And if you look at some of the claims in these articles, it says here, the CLP model closely replicates the clinical picture of sepsis encountered in human patients. So some people have taken this model and now they give antibiotics. And here's another statement. Animals are given antibiotics postoperatively to mimic management of the human disease, as fluid resuscitation and antibiotics are the mainstay of early sepsis treatment. So this model allows fecal peritonitis, abscess, and intestinal necrosis to be treated without source control ever. It's as if they're saying, source control, we don't need no stinking source control. This is the perfect model of gross medical negligence and medical malpractice. And it serves as the absolute gold standard for sepsis research today. So I had Monica Kraselek, who's a second-year resident who spent 2 years in my lab. I said, I want you to take the cecal ligation and puncture model and treat it like we would treat a patient. And she did it. And she did an excellent job. And she used the most severe form of this model and started treating the patients well after, the mice rather, treating the mice well after they developed sepsis. And as you might, and shockingly, this has never been done. And when we did it, not unsurprisingly, all the animals survived. And she washed the abdomen out, gave antibiotics, resuscitated them. I mean, you know, who are the patients that are actually dying today of sepsis in our ICUs? Are they these patients? No doubt patients get transferred to our hospital. There's been a delay in diagnosis and they don't have source control. No doubt. They're just not most of the patients that we think about that we can't get a handle on their organ failure. So to quote Box, essentially all models are wrong, but some are useful. And I've had Gary on paraphrase that because he says this to me often. He's a member of this society and a computational modeler. He said, all models are wrong, but if they inform biology, they can be useful. So we need to be mindful of that. While I'm poking fun at our immunologists, they've done a good service to advance our understanding of the biology of the immune system as it responds to infection. But we need to move beyond that. And we need to keep in mind that bacteria are omnipresent in our critically ill patients, and they continuously influence their outcome. I want you to leave with that message. Even when you have a normal CAT scan and negative cultures, bacteria are still influencing the outcome of that patient. I hope I convince you of that. And it's this triangulated response between the host and the microbe and the microbe, and then back from the microbe to the host. And there are all sorts of signals going on between them. And our microbiota and host cells are in a silent but constant bidirectional molecular dialogue with one another. And you need to keep that in mind. And this silent molecular dialogue was best sort of quoted by E.O. Wilson, who said, in war, and nature is a battlefield, one needs secret codes. And we should keep in mind that chemosignals are emitted, sensed, and responded to at all scales of life. E.O. Wilson was a biologist at Harvard. He coined the term sociobiology because he talked about how ants behave in social groups by emitting pheromones and engaging in synchronized activities. And we humans do that also. This is a study published in Science. I don't know if any of you had a chance to see it, but they collected tears from females watching a sad movie. And they put those tears on a little sponge and stuck it under a male's nose. They then did two things. They measured testosterone level, ouch. And then they stuck these males' heads in a functional MRI and showed them sexually explicit pictures. And they were able to suppress areas that they were imaging. And they did beautiful controls where they had just a saline droplet on the woman's cheek and collected that, et cetera. And it was published in Science, so what can I say? But it really tells us, as Jorge Luis Borges said in 1950-something, everything touches everything, and it's true. And as we heard this morning, even among trees in the forest, there's communication underground, and there is a silent conversation going on in nature that we don't understand fully. And bacteria are no exception. They literally intercept signals released by the host during stress. They talk to each other through a system called quorum sensing, and they've evolved their own internet, if you will. And some people call this bacterial socio-microbiology. How does this work? Well, you may not appreciate this, but bacteria are constantly gathering, processing, and transducing host signals, and responding in a context-dependent manner. When that patient comes into your intensive care unit, they are literally altering their own microbiota and any pathobiota that they acquire through that hospitalization. That fundamentally changes the phenotype of those bacteria, such that there's this bacterial-mediated immune engagement at the mucosal surface, be it the lung or the gut. I'm mostly interested in the gut because the sheer biodensity of bacteria along the 200-meter square of the gut offers a better opportunity for this molecular conversation. And then you have what I call this iterative loop of reverse causality. Not only are the microbiota affecting the immune system, but there is reverse causality that immune change is now affecting the microbiota. And the final output of that conversation can be in many directions. You can have molecular detente, you can have blitzkrieg, rapidly evolving sepsis, or guerrilla warfare, where you have this chronic, slow, kind of like a transplant patient, that are immunosuppressed, where they just linger, and 30, 60 days later, they're in multiple organ failure and very difficult to get out of that particular circumstance. So 20 years ago, we did this, and there's your friend, Pseudomonas aeruginosa, gives everyone in this room heartburn and tenesmus when they see it on a culture result, because we know our patients don't do well. And what we did is we made a reporter strain by inserting a plasmid into Pseudomonas aeruginosa that made it turn green when its quorum-sensing signaling system was turned on and its virulence was turned on. And then we placed it into the GI tract via direct cecal puncture into a mouse that was injured with a hepatectomy, an otherwise recoverable injury, or not. And then recovered those strains 24 hours later, and we could see right away that the pseudomonads inside the stressed gut, in the injured gut, they sensed something was wrong with their host. They became what I call in vivo activated to express virulence. And they clustered and adhered to the epithelium, which we did not see in those that we recovered from the control group. And we recovered just as many from both groups. It's just that you don't see them there because they're not turned green. So we designed a bunch of very clever experiments to show that that very reason that those mice die is because of the activation of virulence. And we did it this way. We injected these strains of Pseudomonas, same genotype of strain, into both groups. And then we harvested those strains by taking the cecal contents and placing it on Pseudomonas isolation agar. So the only thing that will grow is Pseudomonas. And then we went ahead and did what we call this intraperitoneal cross-transfer. We picked those strains up, same concentration in each group, and then injected them into sham-operated animals' peritoneum or hepatectomy peritoneum. And as you can see here, right away, the hepatectomy pseudomonads killed the sham-operated mice who got injected with that strain into the peritoneum, as did it in this transfer, but not here. You take the same number of Pseudomonas from a sham-operated animal's cecum, same number, same strain, and then you inject it into the peritoneum of a sham-operated, nothing happens. They don't die. And you had some death here, but it was basically negligible. Their virulence was upregulated. They exhibited much more motility in the peritoneum. They spread faster. And it fits in with this Bass-Becking hypothesis where he said every gene is everywhere, but it is the environment that selects. Think about your patients. Every patient is different. Every ICU is different. Every hospital microbiome is different. Every doctor, every antibiotic that are used in these hospitals are different. What are we actually doing to shape the virulence of those different organisms that are in our patients? And we even saw a morphotype difference in the dish, where some were smooth and some were wrinkled. And when we picked those out, the smooth had no effect, where the wrinkled killed all the mice. And you should conclude from this that microbial phenotype, shaped by the intestinal environment of stress, not bacterial species, not immune background, caused death in this model. This is a study where we took those same strains, we made them red by using a red fluorescent protein, exposed them to morphine and fentanyl and dynorphin, and only the morphine-treated pseudomonads turned red and clumped. We then implanted a subcutaneous morphine pellet in mice and then injected Pseudomonas into the cecum. And if you look and did all the right groups, you had all the controls, with and without it, placebo pellets, the usual thing. And you can see here that in the epithelium, it's intact, in a placebo-treated, no Pseudomonas exposed. And that blue coloration in the mucosa is Alcian blue. It's a mucus stain, goblet cell stain. If you add Pseudomonas to this animal model, it makes more mucus, because mucus is a defense factor. The lungs see Pseudomonas, the gut sees Pseudomonas, it secretes a lot of mucus under the MUC2 gene. But the epithelium remains intact, the animals don't die. Same with morphine. Morphine is a mucus secretagogue. Look at all those goblet cells in the H&E stain. There's more mucus there in the presence of a morphine pellet alone. But look what happens when you do both. Where are the goblet cells? Well, it's from the Pseudomonas. No, the Pseudomonas increases it. Oh, it's from the morphine. No, the morphine increases goblet cell. Where did it go? It turns out that all those mice died, and the morphine itself shifted the intestinal Pseudomonas to a mucus-suppressing phenotype. So what we do to our patients dramatically shifts the phenotype of these bacteria and hence the immune system. And maybe what we're measuring on the immune side is a result of what we're doing at a more proximate level to the microbiota and the pathobiota. The virulence of Pseudomonas was in vivo expressed in this model. We showed it with our GFP tag strains, and bacterial translocation played no role in the mortality. Now, this is a busy slide, and I apologize for it, but it took another 15 years to grind out the molecular details of everything that we saw. That's a Pseudomonas in the middle there, bacteria, and on the outside are the epithelium of the gut. And what we showed is things like interferon gamma, opioids, adenosine, they bind to specific receptors and transcriptional factors within on and within Pseudomonas aeruginosa. They activate its quorum-sensing signaling circuitry, and then they cause microbe-driven pathoadaptive responses. And I want you to remember one thing, is that when a Pseudomonas is turned on by environmental cues or host cues like this, it releases things like HQ and O. It's a unstable nitrate that kills all the lactobacilli around it. So these are highly competitive bacteria, and they're successful for a reason. They know how to get rid of their competitors. And this kind of fits into the Red Queen effect, which was coined at the University of Chicago, is that critical illness itself drives the evolvability of the emerging pathobiome. Lee Van Valen stated, organisms must constantly adapt, evolve, and proliferate, not merely to gain reproductive advantage, but also simply to survive, pitted against ever-evolving opposing organisms in an ever-changing environment. Now that underlined text should resonate with everybody in this room, and that is critical illness. So we started looking at the membership and behavior of some of these pathobiomes in critically ill patients, and we published this several years ago. Paul Wischmeyer and Mike Morowitz have followed up on these studies and have done some very nice work in this regard, showing, again, that there's major dysbiosis in the guts of our critically ill patients. But just by way of defining the terminology, we now have great technology to measure all these things: microbial metabolites, proteins, host metabolites, whole proteins in a small sample. And ecologists have been doing this for decades. It's now just coming to light in critically ill patients and in modern, what we now call precision medicine. And you get these beautiful displays, and you can even instantiate the host proteins and metabolites with the bacterial proteins and metabolites and draw beautiful interactome maps. So here's a 40-year-old patient from that paper that was dying of what I call non-resolving organ dysfunction syndrome. I'm not happy with multiple organ failure or sepsis because I think there's a lot of baggage associated with those terms, so I'm going to give you a new term here. It's probably equally as vague as the other ones. But this patient, we started sampling. Look at the antibiotics this patient was exposed to. I mean, you don't look at this because you kind of just take care of your patients, but if you look back at a patient that was in the ICU for a month and saw all the antibiotics they're exposed to, it's shocking. This patient got diflucan and ampho-B because he became candidemic. As you can see, even when we sampled him on day one, we did this by both culture and 16S and 18S sequencing. He still grew Candida or had Candida albicans in his gut. And then by the second time, he had that 4-pathogen community on sample point 2, consisting of C. albicans, S. marcescens, E. fecalis, and K. oxytoca. And we started studying that community. Like, wow, what a weird community. It's a pathobiome that sort of coexisted in this patient. And I don't like to call those healthcare acquired pathogens. They're adapted pathogens. They've changed their phenotype. And so here we are again with diversity. This term keeps popping up both in the public, political lexicon as well as in medicine. Diversity is important at all scales of life. We're learning that, and it's important because you lose microbial diversity once you're in an intensive care unit. And that particular patient lost all his diversity. They had no good guys in there. He had only gamma proteobacteria, which are mostly Enterobacteriaceae. And we cultured that stool, and that stool grew out multi-drug resistant S. marcescens, which was a CRE, it was a carbapenem resistant Enterobacteriaceae. It was a bad guy, it was highly virulent. And we started studying this 4-pathogen community. How did it survive in that gut? And what is it doing? Now, just a quick shout out to our Japanese colleagues, both at the Hokkaido University and at Osaka University, who in 2006 described this. They showed remarkably that when patients came into the emergency room with a stroke, an MI, a burn injury, or shock, before they got antibiotics, 6 hours later, their microbiome had dropped 95% in abundance, 95% in diversity, and when they measured metabolites like short-chain fatty acids, they had dropped in concentration by 90%. 6 hours later, what happened? What's the evolutionary reason behind that? But when you look at these studies, and many more are going to come out very soon, you start saying, well, yeah, sick patients, you give them a lot of antibiotics because they're sick, and they're on antibiotics for a long time because they're sick and critically ill, and of course their outcome's going to be worse. That's not really the point. So in order to test the hypothesis that you need your microbiome to survive critical illness, you got to put it back into the system. And you probably need to put it back into the system in a model in which the animal is harboring the same kind of pathogens that our patients harbor. Because no two pathogens are alike. No two isolates of that pathogen are alike. Their environment shapes their phenotype, and their community structure shapes how they behave with one another and to the host. So here's the model that I'm going to show you in the last few minutes here of my talk that I think is really interesting. And we asked this question: can a fecal transplant rescue mice from lethal gut-derived sepsis due to healthcare adapted pathogens? So we redeployed that same animal model of ours. We did a 30% hepatectomy. Now, if you do a 30% hepatectomy to mouse, they all survive. They do perfectly fine. You can starve them for a day or two, do the liver resection. Nothing happens to these mice. They're very hardy. But if you put those 4 pathogens in the cecum at the time you do the hepatectomy, they die. They have about a 60-70% mortality. And if you give them antibiotics, you have a higher mortality rate. Interesting. So we did this study where we gave them a fecal transplant, pooled normal mouse cecal contents, or we autoclaved it and gave it as the control, the vehicle control. And most of these studies, this is the first one that I'm aware of, just give water or nothing. But we said, no, we got to control for perhaps the nitrogen or carbon that's in that fecal transplant. And we did that. Now, we did it at a time when these animals were already septic. And how do we know that? We sacrificed a group of them, measured blood, liver, lung, and spleen cultures, and they're growing the same organisms that we put in there. These are 4 killer, multi-drug resistant, human healthcare adapted pathogens. And we repeated that enema on day 2, and then looked at, again, diversity, and these are simple representations of what's growing in there, but you can see that the microbiome collapses very quickly after that model. You get a pathobiome in the group treated with an autoclaved fecal transplant. But if you give the real deal back to these mice, and this is by enema, you restore the core microbiome where you have Bacteroidetes, you have lactobacilli in there, you got diversity coming back. And here's what happens to the mortality in the group that got autoclaved or nothing. And here's what happened when you give the fecal transplant to the group. You gave the real deal to the group that got the pathogens. Near 100% rescue from already established systemic sepsis. Now, I spent a lot of money doing whole genome transcriptomes. And we took the liver, spleen, and cecal wall at various time points and had our genome center measure all the genes in there, 25,000 genes in each organ. And we got a lot of information back, instantiated it into the Ingenuity pathway analysis platform, and we think we really understand how a fecal transplant can drive what I call a recovery-directed immune response. And I'm not going to go through all this with you, but in a autoclave-treated animal, you can see genes going up and down in all 3 organs, but they hardly move. And this is on post-op day 2, before the animals recover. So they got the fecal transplant on day 1. They're still sick on day 2. You don't know which ones are going to recover and which ones are going to die. But if you look at their gene arrays, you kind of can see that the FMT is doing something powerful to hold those genes in a stable expression pattern. And if you look at it this way, you can see genes going up and down, but in the fecal transplant in the blue, they're all steady as she goes. So what is missing in models of sepsis? We haven't humanized the mouse gut to get rid of its normal microbiome and harbor a pathobiome, just like every one of our patients. And we need that pathobiome to reflect the life histories of human critical illness. Because we always worry about these pathogens that are lurking in our patients. But what is their life history? Which patients have they passaged through? Whose hands have they been on? What kind of nutrients have they experienced as they're passing through? I'm not going to go through all this because it's really long and I only have a few minutes left, but this is a Shannon diversity index, which is a measure of how much diversity you have in your microbiome, and these are cecal samples on day 2 in those groups of animals. And you can see on day 1, you lose diversity, but it starts to come back in the groups that are treated with a fecal transplant. Not the case when you get an autoclave transplant. The panels in the middle, C, D, and E, are oligotyping. You have to sequence down to isolate, which we did because we sequenced the 4 pathogens. And we could tell right away that our pathobiome started displacing and growing and immediately proliferating in this microbiome. Now look at the cytokine levels. They go up in the autoclave group and down on post-op day 2 before the animals have declared whether they're going to live or die. So, who's in control of the cytokine network? Okay, last few slides. We asked this question. I didn't think it would work. I said, well, it seems to me that the fecal transplant given as an enema is driving a systemic immune clearance response. So let's give the pathogens intraperitoneally, systemically, make the animals sick, let them get septic. It takes about 8 hours and they look very septic. And let's see if giving it in a completely different compartment will rescue these mice. So the idea here is that a fecal transplant will shift this pathobiome to a microbiome. And as immune cells passage through the gut, they become educated not by a pathobiome, but by a microbiome. And this happens. There's very good evidence that your immune system is constantly going in and out of your organs, your lungs and your gut, and sampling what's there and altering its immune program based on the pathogen receptors on these epithelial cells. And so here's the model. Now, those of you in the back that are standing up, you might want to sit down. This might make you dizzy. This might shock you to the core, okay? But in 100% of those mice with systemic sepsis, we could recover them with a fecal transplant. These are multi-drug resistant human pathogens from hospitalized patients to complete wellness. The mice are perfectly fine. We've done this experiment 10 times. No antibiotics, no fluid resuscitation. You can rescue mice, 100% of them, not with an autoclave fecal transplant, although it has some effect, but with a live fecal transplant. Peritoneal cultures in these mice demonstrate near total clearance of the healthcare-acquired pathogens of the peritoneum, of the IP-injected mice treated with the FMT, and those pathogens persist in the mice treated with an autoclave. How is this possible? We think we know because we have all the genetic data and we've done the homework, and we're pretty sure that a fecal microbial transplant drives a recovery-directed immune response at the systemic level. So where did this fecal transplant idea come from? Well, as a surgeon, I have to toot my horn, of course. Now, this is important because it's in Wikipedia. Okay, so it's got to be important. And there he is, Ben Eisman. He was the chairman of surgery at the University of Colorado, was a visiting professor when I was a young man with hair as a resident. And he took his team of surgeons, treated 4 critically ill patients with fulminant pseudomembranous colitis before they knew it was C. diff. And he did it by giving an enema. So, to quote Tolstoy, healthy microbiomes are all alike, but unhealthy pathobiomes are unhealthy in their own special way. A hypothesis to be tested. Remember, a hypothesis is not to be believed. A hypothesis is to be tested. Is that if we maintain the gut microbiota, the rest of the body's organs will behave homeostatically. Okay? And you can measure all you want, proteins in the blood, urine, proteomics, metabolomics. But I shamelessly stole this quote from this book. According to quantum physics, no matter how much information we obtain or how powerful our computing abilities, the outcomes of physical processes cannot be predicted with certainty because they are not determined with certainty. Instead, given the initial state of a system, nature determines its future state through a process that is fundamentally uncertain. Your intensive care unit's microbiome at a given time that you put a patient in there is uncertain. The stochasticity of the response of that patient is uncertain. What microbiome or pathobiome is going to associate with that patient, what immune response they have, what their genetic background, it's uncertain. And emergent properties arise. So the pathoadaptive response during critical illness may be a function of ecological collapse within the microbiome and colonization by emergent pathobiomes that drive and are driven by ever-evolving opposing organisms in an ever-changing environment. So Jorge Luis Borges, everything touches everything. We cannot account for everything that touches our patients. Did our patient take a Z-Pak a month before they came in with their Pseudomonas pneumonia? Did the patient whose hip was replaced, who now has bacteremia and is an ARDS, what happened to them beforehand that put them in that situation? If you want to apply some of this technology to your clinical research, it is now possible. Much of the analysis that we do can be outsourced. I want to just point you to our Microbiome Center. We are now affiliated with the Marine Biology Lab. The University of Chicago manages that laboratory. And we're running a course, Dr. Jack Gilbert and myself, in Woods Hole, Massachusetts in May, where we are going to walk you through this entire process, and you can literally take samples from your patients and get this kind of information. I again want to thank Dr. Pauline Park for the great honor of suggesting my name for this talk, and how honored I am to be able to speak to you as the Max Harry WEIL lecturer. Thank you very much.